Hello everyone, welcome again to my channel. I am Anuradha Kashyap, your biology mentor. Today in this video, I am going to share a topic that is about the RH incompatibility, and that topic is given in your NCERT 11th class in the chapter that is body fluid and circulation. So this topic is very important for the students who are going to prepare for their NEET exam 2025 or in future. So the students who want to know about the RH incompatibility, so this video is for you. So before going towards the RH incompatibility, we have to know about blood. So what is blood actually? Blood is a specialized connective tissue and it is composed of two components. One is your formed element which form the 45% of your blood, and the second component is your plasma, which is whitish in color, slightly yellowish in color, and it form actually for 55% of your blood, right? So blood, plasma and blood formed element, these two things are going to make your blood, which is a specialized connective tissue. In the blood formed component, blood formed element, there are further three things. One is your RBC, second one is your WBC, and the third one is your platelets, right? So RBC is biconcave shape and it is very high, it present in very high amount in your blood. RBC is very important for your blood because it helps in the transportation of the gases in your body, like if we told about the O2. So O2 from the environment towards your body tissue is transported through the RBC, and the CO2 which is formed inside your body tissue, it is transported from the body tissue towards the environment by this RBCs. So this helps in the transportation of the body gases, or we can say O2 and CO2, which is very important actually. And this is explained by the oxygen dissociation, oxygen hemoglobin dissociation come into the breathing and respiration chapter, right? So today in this video I will let you know about only the RBC and related concept, that is RH incompatibility, right? But you have to know about the WBC, which act as a soldier of our body, helps in the activation of the immune system of the body to fight with the foreign pathogen. And the third one is your platelets, which help actually in the coagulation of the blood, whether it is your internal bleeding or external bleeding. For the coagulation of the blood, it is very important component that you have your platelets in your blood, right? So coming towards again the RBC. So you know about the RBC and the blood grouping system, right? Carl and Landsteiner discovered the ABO blood grouping system. According to them, that if RBC consists of different kind of antigen, according to that you have your blood group, right? For example, let us consider that RBC have A antigen. Actually, what is A antigen? It is a kind of special sugar which represent the A antigen, right? In actual what happening, RBC have a kind of antigen which is known as H antigen, and over the H antigen there are another kind of sugar. If it is representing A kind of sugar, then it is known as A antigen, and if it is representing B kind of sugar, then it is B antigen. Actually, there is no A sugar, there is no B sugar. There are different components of sugar, like it is your glucose, mannose, or lactose, we can say, right? So if accordingly different kind of sugars are attached to the H antigen, that are going to represent your blood group, that is A blood group. Whether it is your A blood group, whether it is your B blood group, it is AB blood group, or if there is no antigen, then it is O blood grouping, or we can say zero blood grouping. So you know about the four kind of blood group, that is A blood group, B blood group, AB blood group, and O blood group, right? A blood group is when the A blood group, actually when the A antigen is present over the H antigen, then A blood group. B blood group is when the B antigen is representing over the H antigen, then it is your B blood group. And if A and B both are representing over the H antigen, or we can say over the RBC surface, so it is going to represent your AB blood grouping. Actually, it is an example of codominance, because both antigen are going to represent themselves. Neither A antigen is dominant or nor B antigen is dominant, right? So it is a best example of codominance, in which both the antigen are going to show their dominance. That's why it is a codominance example. And if both blood grouping, if both the antigen are not representing themselves over the RBC surface, then it is your O blood grouping, or we can say zero blood grouping. So actually you have only two kind of antigen, whether it is your A antigen or B antigen. If only one one antigen is present, then A blood group. Like if it is A, then A antigen. If A antigen is present, then A blood group. If B antigen is present, then B blood group. And if AB antigen, both antigens are present and then they show codominance, then it is your A blood grouping. And if both the antigen are absent from your blood, then it is known as zero blood grouping, right? Or we can say O blood grouping. Correct? So you know about the AB and O blood grouping. But what about the positive and negative blood grouping? So this positive and negative blood grouping is determined by a different kind of antigen that is known as Rh antigen. Suppose if there is one RBC and over the one RBC there is A antigen, right? So you will say that your blood group is A, right? But you will say it positive, A positive blood group only when with A antigen, or we can say with A kind of sugar, you have Rh antigen also over the RBC. There is one RBC, and if it is A antigen present over the one RBC, then and Rh antigen is also present over the RBC, then it is your A positive blood group. If only A antigen is present over the RBC and not Rh antigen is present over the RBC, then it is then it is A negative blood grouping, right? I'm explaining it again. Suppose we have one RBC and over the one RBC there is B antigen, right? Over the surface of the RBC, that is B antigen is present. So this is your B blood group, correct? But whether how you will decide that it is positive or negative, there is another kind of antigen which is known as Rh antigen. If Rh antigen is present along with the B antigen, then you will say it is B positive blood group. But if only B antigen is present and Rh antigen is not present there over the RBC, then it is your B negative blood group. Clear? So basically, what is Rh antigen? You all know about the A antigen, B antigen, and if both are present, then it is your AB blood group. If only one is present, then that kind of blood grouping is formed, right? If both are absent, then that is your basically what? Then that is your O blood grouping. But what about the Rh? Rh is basically going to decide whether your blood group is positive or negative. If Rh is present along with that particular kind of antigen, then it will be your positive blood grouping. And if Rh is not present, only that kind of special antigen, whether it is A or B is present, then it is known as actually your that particular blood grouping without Rh, or we can say negative blood grouping, right? Rh antigen comes from the rhesus monkey. Actually, from the rhesus monkey, this antigen comes and it is very important during the matching of the blood that Rh antigen should also match. Suppose I am A positive, right? And if I want to transfuse my blood into another person, so it is very important that he or she should be A positive. If he or she should be A negative, then I am not able to transfuse my blood into that person, because it leads to the coagulation of the blood inside that person, right? And leads to death of that person, which is not a good condition. So during blood basically transfusion, it is very important to know that that the two person have same matching of the blood group or not. If they are if the blood group is matching, then it becomes important that yes, you have to know that it is positive or negative. If like I have A blood group, right? And the person another person also has A blood group, but he or she is A negative and I am A positive, then it is important that we cannot transfuse the blood into the other person, because it leads to the coagulation of the blood. So that's why it is important to transfuse the blood. It is very important that along with the A, B antigen, you have to know about the Rh antigen also, whether it is present or not. Otherwise, it leads to the complications. And I want to let you know about one thing also, that if I am telling that there is A blood group, right? Suppose I am A positive, right? I am A positive means I have I have A antigen as well as Rh antigen over the surface of RBC, right? But Why B antigen is not present in my blood? So actually what happens, B antigen is not present in my blood due to the formation of antibodies into the plasma against the B antigen. I'm repeating it again. Like if I am A positive, so my RBC consists of A antigen plus as well as RH antigen, right? But my but over the RBC into the blood, there is no B antigen. Why B antigen is not present? Because in the plasma, which is another component of your blood, in the plasma there are antibody which act against the B antigen. You know very well that antigen and antibody complex formation when occur, then it leads to the death of the antigen, right? So basically, if there are antibody present in the plasma, then that respective antigen will not present over your RBC. I'm telling you again that if the antibodies are present in the blood plasma, then the respective antigen will not present in your will not present over your RBC. So if my blood grouping is A positive, so the A antigen is present over my RBC, so means there is no antibodies into the plasma of my blood, right? And B antigen is not present over my RBC. That's why B antibodies are present into the plasma of my blood. That's why B antigen is not growing. B antigen not growing because antibodies are present there. But if there is no antigen, if there is no antibody into the plasma, no A antibody, no B antibody, then both antigen are going to represent itself over the RBC. Then that blood group will be AB. And if both kind of antibodies are present into the blood plasma, like if A antibodies and B antibodies, both are antibodies are present over the blood into the blood plasma, then there is no antigen over the surface of RBC. There is no antigen, there is no A antigen, no B antigen. That's why we can say that that person is O blood group, right? If RH antigen is also present and RH antibody is not present into the blood plasma, then RH antigen are going to represent over the RBC surface. That's why the person will O positive. And if the RH antigen is also absent because RH antibodies are present in the blood plasma, then that person become O negative. That means that person doesn't contain any kind of antigen because all the types of antibody are present in their blood plasma. So it becomes a very important criteria for you to know that if the respective antibody will present in the plasma, then the respective antigen will not grow over the surface of the RBC, and that leads to the formation of that special kind of blood group, right? So now let's talk about the RH incompatibility. Actually, what is happening here, there is a couple. Let's suppose there is one couple, that is mother is we can say that mother is RH negative, means the female who is conceiving, or we can say who is pregnant. So that particular female is only having A antigen, whether it is A antigen or B antigen, but she do not have RH antigen. That's why she is RH negative, right? But the father, the person who is the husband of that female, so in that case that person is RH positive. So there is one couple in which male is RH positive and the female is RH negative. I think that criteria is completely clear to you, that RH positive male is here and the RH negative female is here, right? So we all know that the RH positive blood grouping is a dominant character over the RH negative female, over the RH negative character, right? So RH negative female is recessive here and the RH positive male is dominant here. When they are going to become parents, so in that condition the baby will be RH positive because dominant character will always express themselves into the next generation, right? So in the F1 generation you will get the child, and in that and that child will be positive, that is RH positive. But that child or that zygote, that embryo that embryo will be into the mother womb, right? Mother is RH negative. So there is a scenario that there is a womb, and into that womb there is a child, right? There is a embryo or child which is RH positive. Embryo or child is RH positive, and the mother womb is protected by the RH negative blood. Clear? Mother is RH negative means mother have antibody, RH antibody into their blood. That's why that female is Rh negative, because Rh antigen is not present over the RBC. Because if Rh antigen present over the RBC, that means there is no antibody in the blood plasma. But I'm saying that Rh antigen is absent from the mother blood. That means Rh antibodies are present into the blood. But the child who is present into the mother womb, now he is Rh positive. But there is a barrier between the mother blood and the embryo blood, right? How it differentiate from each other with the placenta. There is a barrier, that is a placenta. Placenta act as a barrier here, and in that condition placenta will stop to mixing the blood of the mother and the child, right? So first child will be Rh positive, and the mother blood will be Rh negative. But there is no harm to the child, because both the blood are separated by a well differentiated plasma. So here the first child will be completely cured, we can say completely actually in good condition. So when there is a delivery of the first child, during that condition, if, remember my word, if there is rupturing of placenta at any condition, so during the delivery of the first child, if there occur rupturing of the placenta, then there is a chances of the mixing of the blood of child and the mother. Clear? Into the mother blood, there is Rh antibody already present, because mother is Rh negative, right? So in that condition, Rh antibody will be present in the mother blood, and when there is mixing of the blood of child and mother, so baby is actually what? Baby is Rh positive, and in that condition Rh antigen is present over there, but Rh antibody are present into the mother blood. So as the first delivery occur, first child will be completely safe, but the mixing of blood occur into the mother blood. Clear? So as the baby antibody, Rh antibody get fused with the as a baby Rh antigen gets fused with the mother's Rh antibody, which occurring into the plasma of the mother blood, so there is coagulation. There is a formation of antigen antibody complex. Rh antigen antibody complex occur into the mother blood, right? Now what happened actually due to that? Due to that, as the Rh antigen from the baby goes into the mother blood, there is a formation of Rh antigen antibody complex into the mother blood. This leads no harm to the mother, because in the mother body there is no Rh antigen, but that leads to the progression of the Rh antibody into the mother blood. That's why there are lots of antibody formed against the Rh antigen. That is, into the mother blood there are lot of Rh antibodies now. Lot of Rh antibodies are formed into the mother blood, right? So now that is not good actually for the second baby. Now the female is pregnant again, right? So for in that condition, what happen actually? Again, the female is Rh negative, but female, or we can say the mother is Rh negative, and into the mother blood there is Rh antibody in the large amount. Clear? And father is again Rh positive, so again the baby will be Rh positive, because the dominant character will express itself, right? So into the baby is protected into the mother womb, right? But at this time mother blood have Rh antibody in large amount. Clear? And the special feature of the Rh antibody is that it is like to IgG antibody, which can cross the placenta again. So the Rh antibody also cross the placenta. Rh antibody can also cross the placenta. That's a special character of the Rh antibody. So again, this is the womb where child occur, right? Child is bearing. So this is the mother womb, and this is the child into the mother womb, which is protected by a placenta. But at this time, suppose this is the mother blood and this is the child blood, right, into the womb, and in between these there is a placenta. So what happens over the mother blood? Into the mother blood there is large amount of Rh antibody. So now these Rh antibodies can cross the placenta. Now they are going to cross the placenta and reach in towards the baby blood. Clear? Into the baby blood there are RBCs, and RBCs have what? They have RH antigen. Why? Because baby is RH positive, because the character is dominant, right? So now there is RH positive baby and RH antibody cross the placenta. In that condition there is a formation of, or we can say the coagulation of the RBCs. How? There is a formation of RH antibody and antigen complex over the surface of the RBC, which leads to the clumping of the RBC. That's why there is a loss of blood, or we can say a lot of the RBCs will die and leads to the formation of anemia and jaundice into the baby, due to which death of the baby will occur. Clear? So second baby is not safe. Again I'm repeating, second baby is not safe. Why? Because the second baby also shows the dominant character of their father, and baby have RH positive antigen. Baby is RH positive actually, so it has RH antigen over the surface of the RBC, but mother is RH negative. So in that case mother have RH antibody from the first delivery. That's why these antibody can cross the placenta. Now there is RH antigen over the surface of the RBC of the baby, and these RH antibodies from the blood of the mother can cross the placenta and reach towards the RBC, reach towards the RH antigen of the baby, and leads to the formation of antigen antibody complex, which again leads to the clumping of the RBC, and that's why death of the baby will occur. Clear? So actually it is case of RH mismatching, where RH negative and RH negative mother and RH positive father will leads to the death of the second baby. Clear? But how can we control these things? So at this stage you have to know that during the delivery of the first baby, if the mother body is introduced with the RH antibody, or we can say anti-RH antibody, like into the mother blood there is RH antibody now, right? RH antibody now, which was not earlier. And due to the mixing of the blood, RH antibody are high amount into the mother blood, right? So we have to less down their number. So how can we how can we down them actually? So by introducing the anti-RH antibody, we are going to make antibodies against the antibody that is representing RH antibody. So these are the RH antibodies which is into the mother blood right now, and these are the antibody which are against to this RH antibody. Clear? So these antibody will lead to the death, or we can say we can remove them. We can remove the RH antibody from there by helping this anti-RH antibody. And this is introduced into the mother blood during, or we can say at the time of the delivery of the first child, or just after the delivery of the first child, or we can say just before the delivery of the child. And that is known as actually anti-RH antibody, which are very useful for the to safe delivery of the second child, right? And there are rogam also, which is also used to prevent the death of the second baby or during the delivery, because it also helps into the formation of antibody. Rogam, we can say rogam is also available, so it is also used to protect the second child. Clear? So this is a concept you want to know that what is RH incompatibility. Or I want to also say you that RH negative female and RH positive male, the marriage between these two peoples are actually biologically illegal, because we can say that it leads to the death of the second baby. Or if you want to remove the chances of the death of the second baby, so you have to make a gap of at least 5 years between the second delivery, or between the second pregnancy, first and second pregnancy actually. You have to keep a gap of at least 5 years because the antibody which are formed at the time of the first delivery, those antibodies have actually the active phase of their life cycle, active phase of their life at least 5 years. So if you want to remove the chance, or you want to release the chance of the death of the second baby, so you have to keep a gap of at least 5 years between the two babies, so that there are high chances of safe delivery of the second child. Clear? And rogam is one another option. Anti-rh antibody are also another also very good option to safe delivery of the second child. I hope this topic will help you, and this is all about the rh incompatibility. And the students who want to know about more concept like this, so you can comment me down, and please subscribe my channel. And my you can join my link, that is telegram channel. So you want to if you want to join my telegram channel, so I'm giving you the link into the description box, so please join this. And best of luck for your for your exam, and thank you. Stay motivated.